Bismillahirrahmanirrahim. 9700 June 24 question paper 1-3, and this is the first video on that paper. The paper is for 1 hour 15 minutes. Used to be in the previous, before 2022 it was for 1 hour. Now it has increased to 1 hour 15 minutes because the paper is much more difficult now. Question number 1. An eyepiece graticule can be calibrated using a stage micrometer. What is the correct reason why an eyepiece graticule is calibrated? You see, when we calibrate it, then we remove the stage micrometer and then we place a slide, and maybe it's a blood slide. So then we can measure the diameter of the red blood cells or the white blood cells and see if the person is suffering from any disease. So basically, it is used to make measurements. So an eyepiece graticule can be used to make measurements. We remove the stage micrometer, so we once calibrated it. Now we can measure the size of the cells, or we can measure the size of any structure that we want to see if there's any difference between the width of different leaves or the palisade mesophyll cells. How wide is the how much space do they occupy in a leaf from different kinds of leaves. So basically it is to make measurements. Then we come on to the second question. The image is an electron micrograph of a typical eukaryotic cell. What can be concluded about the eukaryotic cell from the electron micrograph? It is an animal cell because it does not have a cell wall. You see, this is the animal cell, and of course it also does not have any vacuole in it. Now this is all wrong. It's an animal cell because it contains a permanent vacuole. That's never been an animal cell. It's plant cell because it contains many chloroplasts. No, we can't see any of the chloroplasts. It's a plant cell, it contains many lysosomes. No, we can't see that either. Question number 3. Which features are found in typical eukaryotes and also in typical bacteria? So, can respire? Yes, bacteria can respire. Contain circular DNA? Yes. So, that's why the answer is C. Yes, bacteria does not have messenger RNA bind to ATS ribosome. ATS is only present in eukaryotes, not in bacteria which are prokaryotes. So, ATS is not common to bacteria. So, this one was wrong. Which type of cell can have the highest proportion of volume taken up with cell structures found by a single membrane? Which type of cell will have the highest proportion of its volume taken up with cell structures bounded by a single membrane? Ciliated epithelial cell, goblet cell, red blood cell, and companion cell. So basically it's going to be, the answer is going to be a goblet cell. Why? Because goblet cells make mucus, and mucus is a glycoprotein. So if it's a glycoprotein, what will we have? We have lots and lots of RER, because the proteins thrown out of the mucus is thrown out of the cell, so it has to be made on the fixed ribosomes, and then it has lots and lots of Golgi apparatus. So highest proportion of table cell structures bounded by a single membrane. What are the double membrane? Chloroplast is a double membrane, mitochondria is a double membrane, the nucleus is a double membrane. Then coming to question number 5, what causes the phosphate heads of phospholipids to become polar? The phosphates become ionized in water. Phosphate heads, how can they be insoluble in water? They are hydrophilic. Phosphates are joined to water molecules by covalent bond. That's also wrong. And this is also wrong. The phosphates are joined to water by hydrogen bonds. Then coming to question number 6. Which statement describes the features of cellulose that are adapted for functions in the plant cell? Cellulose makes up the cell wall. 3 cellulose molecules coil around each other to form a triple helix structure. It's wrong. Many hydrogen bonds form between adjacent molecules. Yes, that's correct. Covalent bonds form between adjacent cellulose molecules. No, that's wrong. So the answer to question number 6 is D, 2 only. Question 7. Which structure shows alpha glucose? Alpha below, beta above. So the answer is B. The rest of them are all wrong. In this we have CH2OH, so this is wrong. In this, of course, the other structures are wrong. This is again. So you got to find out what is wrong in all these three and why B was correct. You got to look at the alpha glucose structure. You have to know it in detail and then find out where things have gone wrong. It's the H and the OH which are not aligned correctly in the rest of them. Now, question number 8. What cannot occur as a result of condensation reaction? What was condensation? Is when water molecule exits. So when bonds are being, when do we need condensation? When condensation means water molecule exits. That means when a bond is being formed. Formation of a disaccharide, joining of two, production of a molecule of water. What cannot occur as a result of? So the answer is A. Breaking of a glycosidic bond cannot occur, in which we have to add water. Then that's hydrolysis. Question number 9. Which fact about the quaternary structure of proteins is correct? Consists of 4 polypeptides, depends on the presence of metal ion, depends on the primary structure of the polypeptides, is made of alpha and beta polypeptides. The answer is C. Depends on the primary structure of the polypeptides. You see, there could be 2 or more polypeptides. So what is their sequence and number of amino acids? That will determine the quaternary structure of it. Because then the secondary structure and then the tertiary structure and then of course the bonds between the different polypeptide chains will occur. Question number 10. The diagram shows different molecules in a solution. Which statement could explain what happens when some of the molecules are mixed together? Molecule P forms an enzyme substrate complex with a non-competitive inhibitor. No, that's all rubbish. They don't form an enzyme substrate complex with an inhibitor. Molecule P binds to molecule P, increasing a bond. Enzymes, please remember, decrease the activation energy. And the C is correct. Molecules R and S bind to the active site of molecule P. Molecule S and R are the products of breakdown of molecule P. P is the enzyme. How can that break down? Question number 11. The effect of substrate concentration on an enzyme catalyzed reaction was measured in 3 different conditions. Without an inhibitor, with a competitive and a non-competitive inhibitor. Rate of reaction 1, 2, 3. Which row is correct? Without an inhibitor. So 1 would be without an inhibitor, 2 would be the competitive inhibitor, and 3 would be the non-competitive inhibitor. So that is very easy question. 11, the answer was A. Question number 12. Which aspect of enzyme activity can be compared by the Michaelis-Menten constant, which is shown as Km? Activation energy of a reaction with a no. Affinity of different enzymes for their substrates. Yes. Higher the Km, lower the affinity. So this is how we have got to understand this. Affinity of an enzyme at different substrate concentrations, maximum rate, Vmax at different temperatures. No, basically, what aspect of enzyme can be compared? So affinity of different enzymes for their substrate. You see, enzyme would only have one substrate. Every enzyme would have its own substrate. Can't be one enzyme, can't have 20 substrates fitting into it. Question number 13. The number of substrate molecules of one enzyme molecule can convert a product in a second is called the turnover number. The number of substrate molecules one enzyme molecule can convert to product in a second is called the turnover number. The number is obtained when all conditions are optimal for the specific enzyme catalyzed reaction. Enzyme catalase 2850 zeros, 28 lakhs, carbonic anhydrase 600,000, phosphatase 971, protease 100. How many times faster at converting substrate to product is catalase compared to phosphatase? So catalase compared to phosphatase. So this divided by 971 and you get 2884, and so the answer is C. Then coming to question number 14, a red indicator solution was mixed with agar and the resulting solid was cut into small cuboid blocks. The blocks were placed in acid which turns the indicator yellow, and another variable is kept constant. Dimensions are given. Which row shows the correct surface area to volume ratio for each block and the time taken from the block to turn yellow? Red indicator solution was mixed with agar and the resulting solid was cut into small cuboid blocks. The blocks were placed in an acid which turns the indicator yellow, and all other variables were kept constant. The dimensions of these blocks used are shown. Block 333, block 2 888, and block 3 11 11 11. Which row shows the correct surface area to volume ratio for each block and the time? You see surface area, how would you calculate that? Length into breadth into 6. So 3 into 3 is 6 and 6 6 are, so you've got 3 3s are 9 and 9 6 are 54. Volume is L cube, so 3 3s are 9, 9 3s are 27. So 54 ratio 27 is 2 ratio 1, so either of these was correct. Now as these increase the size, the surface area to volume ratio is decreasing. Now this and this, all these are the same. What is changing is the time to turn color. So you can see which one, the larger the surface area should take less time. So 4, 11 and 13. So question number 14, the answer was C. As the surface area to volume ratio is decreasing, the time taken will increase. Question number 15. The statement describes some events in the process of exocytosis of a glycoprotein molecule. You know, glycoproteins are made at the Golgi, and then secretory vesicle, and then of course it attaches to the cell membrane, and then exocytosis. So membrane of the Golgi body folds around the glycoprotein. Vesicles binds to and fuses with the cell surface membrane. This should be right at the end. Vesicles attached to microtubule moves through the cytoplasm, secretory vesicles forms. Now basically it wasn't very difficult. Is B. 1 would be the first one. Then 4 secretory vesicles form. Then 3 vesicles attached to microtubules move through the cytoplasm, and 4th would the vesicle bind and fuse and with the cell membrane, and then exocytosis takes place. Now coming to question number 16, 4 cylinders that were identical to size A, B, C and D were cut from potatoes that had been stored for different lengths of time. The cylinders were weighed, immersed in 10% salt solution for 45 minutes and then reweighed. The percentage change in the mass was calculated. Which cylinder had a water potential similar to the 10% salt solution? Now we've been given some figures. Percentage change in mass A was minus 7.2, minus 2.5, minus 0.9 and plus 3.4. Which cylinder had a water potential similar to the 10% salt solution? You see in this question you had to see is which would have the minimal amount of percentage change in mass, because you see there should be, it said it was similar to the 10% salt solution. Of course there should be zero change in mass, but here of course the least change in mass was minus 0.9, so the answer would be C, because the rest of them has a lot of change in mass. Whether it's negative means losing mass or would it be gaining mass. So which cylinder had similar to the 10% solution? So whichever one has the least change in mass. So question number 16, the answer is C. Then coming to question number 17, the contents of a daughter cell are compared to the parent cell after one cell cycle. Which row is correct? Now the question is a one cell cycle, so one daughter cell is compared, so there are two daughter cells now. The main thing is that the cytoplasm volume would decrease. So the chromosome number remains the same, the volume of cytoplasm decreases, the length of the telomeres will also decrease. So the answer is D. That's what happens after every mitotic activity, and that's what causes aging. So question 17 is the answer is D. Question number 18, a high power photomicrograph shows a cell in a stage of mitosis. The chromosomes are visible and lined up along the cell equator. Visible, lined up along the cell equator. Middle, metaphase, but there is no nuclear envelope. So the answer is metaphase. Which stage of mitosis is shown by the photomicrograph? Then coming to question number 19, which statements about the cell cycle are correct? The cell cycle includes interphase and mitosis. DNA replication takes place in interphase. A cell can remain in interphase for several months. All of these are correct, so that's why the answer is A. Cell cycle includes interphase and mitosis, that's correct. DNA replication takes place in S phase of interphase, and a cell can remain in interphase for several months, that is correct as well. Then coming to question number 20, a scientist stains the chromosomes of a plant cell and with a fluorescent dye to observe the telomeres. This cell has 38 chromosomes. How many telomeres will the scientist observe in one of the nuclei during telophase of mitosis? Very, very good question. You see, what is a chromosome? This is a chromosome and it has two telomeres. But what the question is that what is happening in telophase? Telophase, two nucleuses have formed. And each would have 38 in each, 38 chromosomes in each. And one chromosome has two, so 38 multiplied by 2, that is 76. So B, 76. How many telomeres will the scientists observe in one of the nuclei during telophase? Telophase, T for telo, T for 2. So one cell has two nuclei. And then two cells will be formed. So two cells will both have 38 chromosomes because this is mitosis. Chromosome number remains the same. And as each chromosome has two ends, it has two telomeres. So 38 will have 76 telomeres. Now coming to question number 21, during the semi-conservative replication of DNA, the double helix is unwound by an enzyme, helicase, we know that. Which diagram shows how the strands are copied? Now first of all, what do we know about that? The leading strand is supposed to be the 5'3" direction. So this one is wrong. The leading strand has to be in the 5'3" direction. So they've made the arrow, this arrow is going from 3 to 5. That is wrong. This can't be made from 3 to 5. It has to be 5'3" direction. So this was wrong here. Now let's look at C and D. Now this is correct. This is 5'3" direction, so this is the correct arrow. This is 5'3" direction, this is the correct arrow. Then this would be the one coming from the replication fork. So this would be the 5'3" direction. So this is correct in this way. This is also correct in this way. But here, what is wrong here? You see, is the labeling. They've called this the leading strand here, and this they've called the lagging strand here. So this is the one. The lagging strand is made in short bursts, and then it's sort of joined together by the DNA ligase, and these are called the Okazaki fragments. So the Okazaki fragments are on the lagging strand. Here they've labeled the leading strand, so this is wrong, and that is why D is correct. Leading strand and lagging strand, you've got to very well understand it. Leading strand is 5'3" direction, so it will be the 3 end of the DNA the original DNA. and the other one is going to be from the replication fork. Question number 22. A transcription error results in the deletion of one nucleotide from the middle of a primary transcript. mRNA forms from the primary transcript. Which statement describes one possible effect of this deletion on the protein translated from this mRNA? Now, you have to look at it, there's a DNA. So DNA says A, anything. I mean, I'm just saying this. So you can say anything. G. Now, one of this is not copied. So here we have U, A, and say this one is not copied. So we have GG, instead of GGG, now we have GGC. This one comes here. So this is now code, maybe codes for the same amino acid, but from onwards now everything is changed. The whole thing is changed. So let's see, there was this here. Now whole thing changes because you see you got the C, and then one is missing, so you get this to be G. So GGC will be the primary transcript. And then we have, what do we have? Then we have GGT will be translated. So it will be CCA. So after this, everything will change. So the answer is D. The sequence of amino acids in the protein will be different as all the codons from the deletion onwards are changed. From the deletion onwards are changed. Question number 23. Which statement correctly describes the process of translation? Now, what was translation? Translation is when the mRNA gets attached on the ribosome, and those are called codons, and then anticodons are on the tRNA. T, anti-T. Anticodon is on the tRNA. The tRNA brings the appropriate amino acid, and then the amino acids are joined to form a polypeptide chain. Now, one, it says the nucleotide sequence on an mRNA molecule is used to produce a specific amino acid chain. Well, that's correct. A section of DNA is copied into an mRNA. That's wrong. A polypeptide produced because anticodons and tRNA molecule attached to mRNA through peptide bond. Now, that is the wrong thing. Peptide bonds is wrong. So answer is C1 only. Then question number 24. A molecule of mRNA was used in translation. Part of its sequence is shown. There were no introns present in the section of DNA that was transcribed to make this mRNA. What is the sequence of the non-transcribed DNA stand for this section? Now, please remember this. I always ask you to do this in writing. So I would do it like this: is that I'd write the mRNA, then I'd write the DNA. But this was the one which was transcribed. We are talking of the non-transcribed, so it means the other DNA strand. So this is the whole. This is one DNA double helix. They were asking for this, the non-transcribed, the one which has not been transcribed by the mRNA. Because DNA has two strands. One is the transcribed, one is the non-transcribed. So that is why the answer is C. But see how I've done it. You see, I would do it in writing. I wouldn't do it just verbally and think, okay, I'll do it. So mRNA GAU, so DNA will be CT. The T is only in the DNA, CTA. And the other strand would be GAT. So that is what they are asking you. What is the non-transcribed DNA strand? This completes this video from question 1 to 24. Thank you very much.